Okay, well, everybody's finishing getting their morning breakfast. Let me get started. I want to start by announcing next seminar for those that are watching or maybe interested. Our next seminar will be on March 19th. It'll be Doug Weber from the University of Pittsburgh School of Medicine. Will be in here to talk on the 19th of March, and his highly descriptive title will be Sensory Motor Neuroprosthetics. So that's where we're at, but he'll be coming talking about that. He does a lot of spinal cord stimulation interaction work. Another announcement I want to make for the NP seminar that's been published or advertised in the book. If you have this on your calendar and marked with stars to make sure you make it, switch it from the 21st of May to the 14th of May. So we've shifted back a week on Paolo Bonato, who's our May speaker. And just to round it out while I'm announcing, on April 16th, Stuart Kogan will be here from EIC Laboratories talking about materials for stimulation. Okay, today I want to introduce our speaker coming to us from Vanderbilt University. It's Duco Jansen. He, by way of introduction, I'll read the biostats first here. The University of Utrecht, or some other way I mispronounced it, but in the Netherlands. Got his Master's of Science in '90, and then his Master's and Ph.D. Culminating in '94 from the University of Texas at Austin. Dr. Jansen basically is one of the founders of infrared neural stimulation that we'll hear a lot more about today. He got in the faculty at Vanderbilt, began his career there in '97, assistant professor, and worked up to basically a full professor in 2008. He's a member of the Vanderbilt University Institute of Imaging Science, also a member of the Vanderbilt Institute of Integrated Biosystems Research and Education. Starting with the Whitaker Foundation Young Investigator Award. He's been in chair of societies like the International Biomedical Optics Society. He's recently the president-elect of the American Society for Laser Surgery and Medicine. He's a fellow in SPE. He's a fellow of the American Society for Laser Surgery and Medicine. And in addition to his research, he's also earned Excellence in Teaching Awards at Vanderbilt. He has over 70-some publications in basically photonics and biophotonics and the work that's related to that. I've been fortunate enough to be more and more involved. I think I'm fortunate enough to be more and more involved recently with looking at new ways. You know, we've been very focused on electrical stimulation interactions here, but it's been very good in the last, more or less, I would say, probably a year, 6 months to a year, that we've been in conversations of how do we kind of combine these technologies and look to bring maybe this idea from a selectivity perspective and refining our protocols for peripheral nerve stimulation. So for me, it's been a very rewarding interaction so far, and hopefully to continue to grow in the future. So without any further ado and listening to me, let's listen to the key interest for today. So I'd like to introduce Dr. Duco Jansen.
Thank you. All right, thanks, Dr. Tyler. Thanks for inviting me. I have to apologize as well. I really wish I could have spent more time. Turns out my wife is on an NIH study section. She was supposed to come back 30 minutes before I left from the airport in Nashville. Of course, she got stuck in the snow, so I dumped my kids off with 2 boxes of mac and cheese, and I left. All right, so I'll talk today about what we've been doing for the last several years in the area of optical stimulation. And to sort of put this in perspective, I'm an optics guy, and I'm starting to learn a lot about the neural system, but you know much more about that than I do. So here's the outline of what I want to talk about. I'll give you a little bit of background on the concept and how, what is this all about? How do we do this? What sort of parameters are important? A little bit on laser development. We're going to have to build the devices to make this happen. In order to get to good devices, we have to understand the underlying mechanisms. And that's not just an academic, it's an interesting scientific question, but it's critical if you want to develop devices that do this effectively as well. I'll talk a little bit about a couple of applications. I won't probably get to all of them, but to kind of give you a glimpse. And I want to end with some what I think are opportunities, and specifically are some challenges that are going to take some really bright minds to move this forward. Just as a disclosure, we're talking about equipment and devices here that are not yet approved by the FDA. We do have some IP on this, and I get some royalties from this company, Lockheed Martin. So how did this all start? A good colleague of mine, neurosurgeon Pete Conrad over at Vanderbilt's Medical Center, came to us several years ago, and Pete is a functional neurosurgeon. He does a lot of putting electrodes in people's brains for all sorts of procedures, and he came to us with the question, you know, is there some way you guys, optics people, can make visible to me what part of the brain is being activated at any point in time? Typically what they do there is they map things out before they put these electrodes in. It's a tedious job. If you do this in July and August, then you have first-year residents in the OR, things have a tendency to go really interesting. And so he really wanted to do this and say, can I somehow look at that? And so we started brainstorming about that. We did some literature searches, and the short answer there was, yes, you can do this. It has been done. There's a lot more that needs to be done. It's not yet, anyway, in clinical practice. But the fact that a lot of people are already working on that with things like diffuse optical tomography, optical coherence tomography, fluorescence imaging, both intrinsic as well as extrinsic signals, you know, it's possible to do that. So to me, it was much more interesting to actually turn that around and say, okay, well, that's great. If you can see electrical activity in the brain with light, can we do it the other way around? Can we induce electrical activity in the brain with light? And of course, then the question is, well, why would you possibly want to do that? All right, this slide I'm going to go over very quickly for this crowd. Neurostimulation, as you probably know, is very important for all sorts of applications, both diagnostic and therapeutic, and it's the main tool in applied and basic scientific research, neuroscience, and electrophysiology. So there's a real reason to want to do this. Interestingly, and despite all sorts of developments in this field, fundamentally, the technologies that are being used for that, using electrodes to induce electric fields to then trigger electrical activity in excitable tissues, really hasn't changed that much in almost 2 centuries. And this goes back, you know, to Fritz and Hitzig, and you know, if you really want to look at it, even to Alexander Volta way back when. Just to sort of reiterate, what are the problems with this? It's a technique that works. It's a technique that's being used for lots of things in the clinic, in patients, in the research labs. There's a number of fundamental limitations here, and that is spatial precision of stimulation. As you all know, when you put an electrode in biological tissue, you put that electric field in. No matter how small you make that electrode, there is a finite electrical field that's induced in this tissue. And unless you physically impale these neurons, I mean, that is sort of a fundamental physical limitation that one has to contend with, no matter how fancy you make your electrodes. The other issue, and this is probably more relevant in the sort of research setting than it is clinically, but anytime you do electric stimulation, recording pairs, you always have this nuisance of a huge stimulation artifact on your recording electrode, unless you go physically far enough away you can separate those two in time. And wouldn't it be nice if we can measure right where we're stimulating? And then lastly, there's always a need for physical contact between the nerve and the electrodes, and that may have its own set of problems. There is the issue of once you have electrode leads, especially metallic ones, in those tissues, you know, what about the compatibility with MRI? And can we do better? So we came up with the hypothesis and say, okay, can we use pulsed laser light to induce activity in a damage-free, artifact-free, and highly precise way in discrete population of neural fibers? So that was sort of our initial hypothesis. We wanted to make sure, we want to validate this, show the feasibility, compare it to electric stimulation. We were really interested in developing devices to make that happen, and then we have a real interest in getting this to more sophisticated systems from the periphery all the way to being able to do this in the brain. All right, so let me define what is optical stimulation. So our working definition of this is basically this. It's the induction of an evoked potential, an EP or an AP, in response to a transient targeted deposition of optical energy. So that's our working definition. Now, if you type in optical and stimulation in Medline or Web of Science, you get all sorts of returns in areas that actually do not have a lot to do with what we're talking about here. And so what is it not? There's a whole field out there of sort of biostimulation or so-called low light level therapies. This is things where people use laser pointers, type lasers to try to modulate biological activity. There's interesting work going on in there, but those are all very low intensity. They're not transient, and you know, they use them for modulating physiology to some extent. Also, there is a whole area out there where you can optically modulate the neural signals activity, so they're induced otherwise or they're intrinsic, and you can modulate them using light somehow. There's some fantastic work going on in the whole area of genetic probes, genetically engineered light activatable ion channels. So these are the bacteriorhodopsins, that work that's going on at Stanford and MIT, for example. And they're very, very elegant reports coming out of that. My personal bias here is that I'm not quite sure how that's going to translate to clinical applications, but you know, perhaps people will come up with a way to do that. Light activation of electric stimulation. So this is sort of, it's an electric stimulator, but somehow you use the light to drive that, but it's still fundamentally an electrode tissue interface that does the stimulation. And then there's an area of caged compounds. These are molecules that have been inactivated and can be activated using light. So those are all things that are out there that sort of fall in this general area of optical stimulation, but really are not what we're talking about here. So let me just start with, what does this look like? And you know, once Pete and Anita and myself, we sat around the table, came up with this idea of, well, can we use light to stimulate electrical activity in tissue? We actually went to the lab and decided to try it. And it's probably the one and only time in my life that an experiment actually worked the first time. In hindsight, we were very fortuitous in picking the parameters that we did. We had at that time at Vanderbilt a free electron laser. Now, a free electron laser is a device slightly different than most lasers. For one, it's about a building worth of size. It's free not in a monetary sense by any stretch of the imagination, but it's free in the sense that we use free electrons to produce light. And what that gives us is a knob that allows us to change the color of light, the wavelength of the light. Any conventional laser that you buy, for the most part, depending on the material that's lasing, gives you the wavelength, the color of light that you're going to get. And if that happens to be the wrong color of light and you need another one, you got to buy a different laser. So with the free electron laser, we had the opportunity to actually experimentally explore the parameter space, and we did that. And I'll come back to that a little bit. So basically what we have here is a little video. So this is a rat, sciatic nerve over here. What you see on the top is a fiber optic that comes down over the nerve. And we're pulsing this laser with, I believe this is 2 pulses per second. And what's really interesting, so this is a non-contact. And what's interesting here is that depending on where exactly we place the fiber, we can induce activity in one specific muscle group, but not have all the other muscle groups react or respond to that. The mapping of that is a story in and by itself, but you know, what this sort of illustrates is that we have this non-contact way of stimulating this tissue with reasonably high precision. Now, the first thing we thought when we did this experiment was, okay, well, even though the animal is anesthetized, maybe it's feeling pain and there's a reflex arc that's causing this. So the next experiment you do then is to cut the nerve upstream to make sure that is actually that link is broken, and when we did the experiment again on the same animal, we still saw these motor responses, suggesting indeed that we're getting a pulse that travels down and invokes this compound muscle action potential. All right, so kind of laying this out now, after we did some initial work with the free electron laser, we found that a couple of wavelengths really worked well, and I'll come back to exactly that in a minute. We had a standalone. This is now a different type of laser. This is not a building, but it's only an optical table full of stuff. couple that into a fiber, bring that fiber in, as you just saw, to the nerve, do our standard muscle recording, nerve recording, and also bring in the electric stimulation to sort of characterize what we have here. And to graphically illustrate what I just showed you in that little movie, standard electric stimulation here, proximal to the branching point, with a bipolar hook electrode. What you get downstream is sort of two things. First of all, you get this big stimulation artifact on both this fascicle and that fascicle, and you get a nice response, but it's not spatially specific. All right. If we do the exact same thing now, fiber optically stimulate this, first of all, you see the lack of artifact, and secondly, you see the signal on one but not on the other fascicle downstream. So that basically kind of hits home the point I just showed in that movie. The other thing that's worth noting is the amplitude of these signals. Here after gain, we get the gain the same. After gain, we're at what is it, a couple of volts over here. Here we're at about 1/10 of that. So that has everything to do with recruiting, right? How many axons do you recruit? And so we're clearly, these are compound potentials, so clearly we're recording much fewer axons optically than we are recording than we're stimulating electrically. So we did some proof of principle there. And since then, this project has grown in a large number of different directions. And this is sort of the roadmap of where things are and how it all ties together. There is some work on the peripheral nervous system. The laser parameter optimization, the functional mapping of these nerves is what we're working on. There is a significant effort underway in developing devices and then making sure that they actually work the way we think they do. I'll talk a little bit about mechanisms of underlying how does this really work, both physically and physiologically. We have a very interesting collaboration with Northwestern University, Klaus-Peter Richter and Aiello Izzo, to effectively build a cochlear implant based on this concept. And we're also dabbling into stimulating with the central nervous system directly somewhat. So I'll hit on some of those things as we go forward. So the first question you really have to ask yourself, once we were lucky enough to make that first experiment work, what is the best wavelength of light to do this with? And this is sort of, as I said, I'm an optics guy. This is sort of my life in a box. From 100 nanometers UV all the way to 10 microns in the infrared, what in biological tissue exactly absorbs light? And if you kind of look at this, there's a couple of critical things here. On the short side here, the UV, it's mostly the proteins, water also. In the visible, it's predominantly melanin and hemoglobin that does the job as far as light absorption. And then further out in the mid and near infrared, it's the water absorption curve that dominates this concept, because after all, soft tissues in particular, it's about 70–75% water anyway. Okay. In a rat sciatic nerve, if you look at the dimensions here, the nerve having a diameter of a couple of millimeters or so, we have the outer sheets, the epineurium we need to get through, we have the perineurium, and we'd like to only stimulate one fascicle but not the others. And so dimension-wise, we got to get through this first 150, 200 microns worth of connective tissue up with light, and then we have to stimulate the thickness of a fascicle. So if you put that together, you come to the conclusion that ideally for peripheral nerves in the rat, we need light that penetrates on the order of 1/4 to 1/2 millimeter into this biological tissue. Stated differently, and I always get caught on this because this is second nature to me, but the optical penetration depth is essentially the inverse of what we have here, which is the absorption coefficient. For convenience sake, we're just ignoring the fact that light also scatters. We'll have to deal with that later, but for now, we'll just play like it's all nicely getting absorbed. So if you look at this and you say, okay, well, what parts of the spectrum are interesting, and what gives us sort of this 250 to 500 micron penetration, optical penetration depth? And in the mid-infrared, there's an area that is particularly attractive. So let's look at that a little bit more detail. So now you have essentially the same graph. It's now plotted as penetration depth and no longer on a log scale. And you say, okay, I need penetration depth sort of in this area between, called 150 and 550 microns penetration depth. That's what I'd like to have. So where does the green light, the absorption curve, fall within the gray box? Now the other interesting thing here is that when we want to make this practical, it behooves us to start looking at how we're going to get that light to this tissue eventually. So fiber optics become our delivery system. And if you look at fiber optics, they work really well up to about 2.5 microns in the infrared. Beyond that, you really run into trouble with that. There are materials out there, but they have all sorts of issues in terms of toxicity and brittleness and what have you. So that kind of limits us to this area. And as I said before, and we did this experimentally with the tunable laser. We kind of settled on this Holmium YAG laser, which has a wavelength of 2.12 microns and a penetration depth of about 1/3 of a millimeter, 350 microns or so. That is an existing laser. It's a tabletop sort of device. I won't call it portable, but it is movable. And so that is an attractive device to do that with. Zooming in on this little peak here, again, this is the water absorption peak. It peaks at 1.93 micrometers of wavelength. The Holmium YAG laser that I just talked about sits here at 212 with this penetration depth, this absorption coefficient, this penetration depth over here. Okay, so that's basically what we did most of our initial work with. As I said, that's a movable laser, but it's not a portable laser. And so, as I'll talk a little bit about a little bit later, we're interested in other laser developments, and so there's no reason why we couldn't go to the other side of that absorption peak, and I'll come back to that laser later to develop standalone small portable devices. So we went from, this is the Vanderbilt Free Electron Laser Center. That's the part that's above ground. The laser is in the basement, and we pipe the beam throughout the building. We did the initial work there. We translated it, figured out we can do this with the Holmium YAG laser, which is this box over here. As I said, it's an optical table full of stuff. And then we really set out to say, okay, can we come up with I want a laser that I can throw in my backpack, that I can plug in the wall and plug and play literally. And that looks sort of like an optical version of a grass stimulator. That's what I wanted. So we started contacting and working with companies, and it turns out that the laser wavelength that I just pointed out here, I mean, if the Holmium works, there's no reason why another laser with the same penetration depth, but on the other side of the water absorption curve, shouldn't work. And so it turns out there is a diode material out there, indium gallium arsenic phosphate, that can do that. And so it turns out that that's a laser development that has been done by the military. And it's sort of the thing that, the sources that form the heart of the infrared countermeasures in the Apache helicopter. So conveniently, the DoD guys have developed the lasers for us, and we put one and one together and came up with sort of the initial prototype of what this thing should look like. As you see, it's got the laser driver and the laser head, so it's a two-piece. So pretty quickly, it became known as the bikini laser. From that initial prototype, we went to, I say we, this was largely done by Acculite, which last year got acquired by Lockheed Martin, but they have now developed this device, which really does look a lot like the optical version of a grass stimulator. So I won't go through the parameters here, but it's a little bit tunable depending on how hot you make the diode, and that's actually beneficial because it allows you a little bit of optical tuning there. But basically, this thing is the size of twice my laptop. You plug in the fiber, and you get similar buttons to what you'll find in a typical grass stimulator: pulse rep rate, pulse duration, intensity, and so on and so forth. So this thing has been built. It is actually marketed and being sold now for research purposes. And since that thing is now available and reasonably affordable, a lot of people have started to work on this technology, and it's actually been a lot of fun for us to sort of see how people have started, different groups have started to pick up on this and start doing different research type projects on this. And so down at the bottom here, you'll see some names that you probably recognize. So we're now working with Andrew Rollins and Hillel Chil on trying to stimulate the heart, and if I have time, I'll show a little bit on that at the end. and also with Hillel trying to understand the mechanisms behind this, using it in aplesia. Okay, so we were convinced that we can in fact stimulate these tissues. We can do it effectively. We can do this with varying parameters. We have some control over this. The real question you then have to ask yourself is, well, are you cooking the tissue in the process? After all, you're shining laser light at reasonable intensities at this tissue, and it's being absorbed. So we spent a good bit of time early on assessing that, and we did a lot of animal experiments, mostly using the rat sciatic nerve as our model here. And we did both acute histology and survival histology. The acute I probably won't talk about, because if you see damage, the threshold for damage visualization is always going to be lower in the survival studies, simply because you'll find the response to damage, which is easier to see than the acute damage. So just to get to some results here. As a function of radiant exposure, we basically did a so-called probit study, where we irradiated this tissue, stimulated it for a set amount of time with a set energy. And the stimulation thresholds are shown over here, okay? You'll see there's a little bit of hysteresis as we go up this curve versus as we go down this curve, but the bottom line is that in the rat sciatic nerve, our radiant exposure that we need to stimulate with a 50% probability is on the order of 0.3 or so joules per centimeter squared. The damage studies, where we basically, and these are the survival ones, so both 3-day and 14 days, we scored them as zeros or ones, so 100% probability or 0% probability, fit this curve to it and come up with what effectively amounts to sort of an ED50. What's the 50% probability of creating damage in these tissues? And you're in around about 1 or so joules per centimeter squared. So we have a margin here. We can do this reliably, repeatedly, without actually damaging this tissue. As I'll show you in a little bit, though, while this is hopeful, I think we can do better, and we're going to have to do better for this to move towards real clinical applications. So the question here now becomes very quickly, and every time I give a talk on this, somebody asks, so I figured I might as well talk about it. How does this work? There's no real reason to believe that these type of tissues, a rat sciatic nerve, should be light sensitive in the first place. There's nothing in there, in these peripheral nerves, that would give them any sort of light sensitivity. So we sat down with our laundry list of, okay, well, how could it possibly work? And being the laser tissue interaction guy that I am, as I said, this is sort of my world. The 4 hypotheses that we came up with is, okay, well, it could be an electric field effect. After all, light is electromagnetic radiation. It travels, transverses this tissue. And what about maybe the electric field somehow magically does this job and does this trick? What about photochemical? Well, perhaps. There may be things going on here that we drive optically. Photothermal is a possibility, and if so, there's a number of options here. And finally, photomechanical. So let's just look through and sort of do a process of elimination here. If this is an electric field effect, what would that look like? Well, the theoretical calculations suggest that this is highly unlikely for 2 reasons. First of all, the electric field strength really is too low to drive activity in excitable tissues that we have. Secondly, and this is perhaps the bigger one, we're dealing with alternating electric fields here with frequencies typically in using light on the order of 10 to the 14th hertz. So based on that alone, it is highly unlikely that that has anything to do with what's going on here. Nevertheless, we decided to just give it a shot, and we swapped out the holmium crystal for an alexandrite crystal, and this is a laser that emits sort of in the near dark red light, 760 in the near infrared, which is essentially not really absorbed in tissue at all. And everything else being the same, we fiber delivered it, we used the same pulse duration, and we just started throwing laser pulses at this tissue to see at what point was where we're going to get these action potentials and these muscle twitches that we were seeing with the Homium. And we were able to go all the way up to about 200 millijoules per pulse and see essentially nothing. To put that in contrast with the Homium, there at 4 to 5 millijoules, we were seeing stimulation. Suggesting that, I mean, the electric field ought to be essentially the same in both of those things. That's with the exception of the frequency a little bit, but that's independent of that wavelength in and by itself. Once we really go over this and we can crank it up so high that we can actually carbonize the tissue, something interesting happens. What happens when you carbonize tissue? It turns black. Well, black absorbs the red light, and it started twitching just fine for a little while. So the bottom line is that we convinced ourselves that this is highly unlikely to be the effect. All right, so moving right along. That one is out. What about photochemistry? So the question is, do axons have unique optical properties? Is there anything special about neural tissue, axons in particular, that would suggest that somehow they would be sensitive to light and to a specific wavelength of light? And so the question is, how do you measure that? Well, it's not so easy because you don't have a lot of tissue, you don't have a nice sheet to stick in a spectrophotometer. So we did a number of different things. We harvested these tissues and froze them and ground them up and spun them down and made pellets and sucrose gradients and what have you. What you basically see and what the take-home point here is, and just for comparison, the purple curve here is just pure water. Okay, looking in this part of the spectrum, for all practical purposes, neural tissues essentially follow along with the water absorption curve. There's nothing really that stands out there that says, okay, there is a specific absorption or a specific photochemical process that might happen at this wavelength or something like that. So the other things to consider here is that we are stimulating now in the infrared where the photon energy is on the order of 0.1 electron volt or less. Most photochemical processes happen in the visible where the photon energy is more like 4 or 5 or so electron volts. So we're an order of magnitude away for direct photochemistry anyway. Now you could still drive photochemistry with this wavelength if you have multiphoton effects. However, the intensities that we're using is 6 orders of magnitude below what you need to get multiphoton effects. So basically, based on that experimental evidence and sort of giving it some thought, it's unlikely that it's that one. So now what? Well, and you see I'm lighting these two up together here, the photothermal and the photomechanical. Why? Because they're intricately coupled and it's very, very difficult to separate those things. As you heat materials, they expand. So right there you have it, they're coupled. So let's see if we can get a handle on this. First thing we did was use the thermal camera to kind of get a feel for how hot does this tissue really get. And this is actually done in a rat. The sciatic nerve is being irradiated with our Homium YAG laser. And this is sort of the cross section here, which you see now. And these temperatures are actually reasonably high. We're getting an average temperature rise here of about 4 degrees Celsius or so. And a maximum temperature rise, given that this is more or less a Gaussian in the center, that's almost double, or that is double that. So it gets hot, or warm at least. So based on that, we can say, okay, well, at least we know it gets warm, and what happens next? I mean, do we somehow, does the thermal effect create transient membrane permeability? Do we change the membranes? Is there something we do to the transmembrane ion channels, perhaps? There's a question you can ask about, well, is it an absolute temperature or is it the temperature gradient? And we did some interesting experiments there. Actually went to a frog model, which is nice because it's a cold-blooded animal. And you can sort of do this in a cold frog or a warm frog and see if the threshold changes. And the short answer there is that it is really a temperature gradient effect. The baseline temperature, the steady state temperature does not seem to really affect the threshold for optical stimulation. I still want to know about this mechanical stuff, and if you look at the interaction of laser light between laser light and biological tissue, there's two important parameters. There's the pulse duration of the laser pulse itself, and there is the penetration depth. I already talked about this one. And what you have to then consider is, well, what happens to this energy as you dump it into this tissue? The penetration depth defines sort of the radiated zone, and the penetration depth defines the irradiated zone, and the pulse duration defines how much time you have to get for energy to diffuse out of that zone. So if you're over here, the heat sort of stays into the irradiated zone for the duration of the laser pulse, so it doesn't diffuse out. It does eventually, but not during the pulse itself. If you're over here, you're what's called stress confined. Now, stress pressures travel a lot faster than heat, and so in order to not have pressures diffuse out and build up significant stresses in this tissue, you would have to be at much, much shorter pulse durations. And so for the lasers that we've used to stimulate, we've always been sort of in this part of the with the holmium laser, we're at this penetration depth, and we've used the holmium laser at different pulse durations. We've used the Acculite laser at 5 milliseconds here, and we've used the FEL at this wavelength at 5 microseconds. So we've been able to span about 3 orders of magnitude in pulse duration. And while they're all thermally confined, this one is getting pretty close to being stress confined. And so what you would expect if there is a pressure effect is that the stimulation threshold for the shorter pulses should be less than the longer pulses, simply because you build up larger stresses here. These lines are not yes/no decision lines. It's a gradual process here. So with that in mind, you can do the experiment and say, okay, what is the stimulation threshold as a function of the pulse duration? And for the pulse durations that I just talked about here, we basically see no statistically significant difference in the stimulation threshold, suggesting that at least the thermoelastic expansion, which is what we're talking about here, things heating up and expanding quickly and causing a pressure wave, that actually plays a negligible role, because if that were a significant factor, you would expect this data to look, somewhat like some slopey curve over there. Now, just because we don't produce these initial thermoelastic pressure waves, when you heat things up, they still slowly expand. And so what about some sort of volumetric expansion? And how much does tissue really expand in the first place when we throw this sort of laser light at it? And so we actually tried to measure that and worked with some colleagues at the University of Texas in Austin. Tom Milner, who has a very nice device. It's a variety of it's a variance to an OCT system with a differential phase option, and it allows us to make measurements of surfaces moving with spatial resolutions on the order of 20 nanometers. And so what we did here is we monitored the surface displacement of this irradiated nerve as we're irradiating it with the laser, and that's basically the curve you see here. And so when we fire the laser, we see an expansion of on the order of 300 nanometers or so, and then it relaxes back to its original position, according to basically this is a thermal diffusion time type constant. So now we know that when we irradiate this tissue with the laser parameters that stimulated, we have a surface displacement of 300 nanometers over some time course that we've defined. We subsequently actually went and built a piezoelectric needle device on which we imposed the waveform that we measured here. So now we have a pure mechanical device without any heat that can do the exact same thing that we did with the laser. And I don't have the data here to show, but because it's all not that interesting, because the result was negative. No matter what we did with that little piezo piston thingy, imposing this mechanical waveform alone, we were unable to stimulate this tissue that way, even with amplitudes 10 times as high as what we were getting with the laser. So that in and by itself suggests that, while we can't completely exclude it, it is unlikely to play a major role in this whole process. So if you put it all together, in all likelihood, it's not this one, it's not that one, as I talked about. There's no pressure waves. There's an unlikely role for thermal expansion. So that sort of leaves you with, in all likelihood, it's a photothermal effect, and that appears to indeed be the driving mechanisms behind this. That still leaves us with the question of, okay, now we have a temperature gradient deposited in this tissue. How in the world does that ultimately lead to opening of sodium channels and creating action potentials? And I wish I had the answer for you, but I don't. We're still working on that. There's a couple of hypotheses that we're currently pursuing. There may be a gradient dependence of the state of the ion channels. We have some preliminary evidence that maybe the chloride channels might be involved as well. There's a hypothesis out there that perhaps the vanilloid receptors, the TRPV channels, might be involved because they are known to be sensitive to temperatures. And there's some elegant work that Klaus at Northwestern has done with that. And so the evidence there is that they might play a role, but probably can't account for the entire effect either. So in all likelihood, this is a complex mechanism that has multiple facets to it, and we're still needing to do a lot more work to really sort that out. Okay, now switching over to some applications here. We asked the question, can we hear light? Which is a really interesting question if you think about it. So just a little bit of background, and as I said, this is really the collaboration, and mostly this is the work that's been done at Northwestern with Klaus-Peter Richter and Agnella Isomatic. So cochlear implants have sort of the same problem that I already talked about. You can only encode so many independent channels of electric stimulation along the basal membrane in the cochlea before you start getting crosstalk to subsequent electrodes and even crosstalk to the next turn of the cochlea. This is sort of what that looks like. I'm sure you all are familiar with that. I just want to get to this cartoon to sort of illustrate that. If you do this with electrodes, the electric field spreads, and you get multiple populations of spiral ganglion cells that might be stimulated by one electrode. Or you might have a situation where two adjacent electrodes stimulate the same population. So this is a problem if you want to encode sound frequencies along the basal membrane. The cartoon for doing this with light, of course, is that we can have some sort of delivery system with the side firing thing in there, like a Bragg grating or something that can specifically stimulate. We have control laterally with light. It's easy to spatially do this accurately. In the actual direction, what we got to do is pick the right wavelengths, as I talked about. Now we can tune the penetration depth by picking the right wavelength. So that's sort of the idea here. So showing that this actually works is done here. So we take an animal, give it an acoustic click, and see what that neural response looks like, both in phase and 180 degrees out of phase. And then we do the same thing with the optical fiber aimed at the spiral ganglion cells. And you see sort of the same idea, but at a smaller amplitude for the reasons that I mentioned before. We're recruiting fewer axons, or fewer ganglion cells in this case. Of course, now we're starting to get into the realm of how long can you do this without damaging or without causing problems in this. And so this is an experiment that was done for 6 hours continuously, measuring the compound action potentials over time. And the little triangles here is when we had to stop the experiments and re-anesthetize the animal. So there's some deviation here. But generally speaking, we don't really see a statistically significant drop-off in the compound action potentials for at least up to 6 hours. Now, 6 hours is a very different story, of course, from a chronic implant. So there's a long way to go there. But at least it looks promising in the short run. This here, and I won't go into the detail of what exactly the experiment entails, but it's essentially a masking experiment that gives you a measure for the spatial accuracy of stimulation. The black dots is the acoustic response. The open circles here is the optical response. And they really line up quite nicely. And what's not shown here is if you do this electrically, this curve looks much, much wider. So proving the point that we have that spatial resolution to do that. That's nice at a couple of hertz. In order to encode sound above the noise floor, we have to go to higher repetition stimulations, obviously. So this becomes interesting because if you put in single laser pulses that cause temperature rises, you could very easily end up in a situation where you get thermal superposition, where you hit the next pulse before the thermal energy is diffused out of the irradiated zone. You could get the temperature ramping up. Now the good news is that the threshold in this particular system for stimulation is 2 orders of magnitude lower than what we had in the peripheral nerve. And it has a lot to do with the amount of tissue we had to go through and the number of axons we actually have to activate to do this. So if the threshold is lower, that means we can get away with throwing a lot less energy at this tissue. And therefore, the heat load overall is significantly less. So here at 200 hertz now, I believe, yeah, this 200 hertz, this is the laser pulses, and the action potentials track quite nicely with that. And here we go all the way up to 300 hertz. You start dropping some pulses every now and then. But generally speaking, it tracks along quite fine. And we don't really have, and this now is at 400 hertz with the action potentials. And so even at 400 hertz with the 15 millijoules per centimeter squared, and again, this number is at least 2 orders of magnitude lower than what I talked about earlier for the periphery. But we retain this response for hours on end, even at 400 hertz. So that's looking promising. So I think we're at a point where we convinced ourselves that this is feasible. There's still a lot of work to be done, obviously. But there's some real promise, and if we can actually do this with spatial resolutions comparable to acoustic stimulation, you may indeed be able to build a cochlear implant that is superior to what can currently be done, at least with electric wires in there. The challenges here is I'm not convinced that we figured out the right wavelength for this application, and that's sort of a take-home message that we've learned as we went through this, is that you really have to tailor the laser parameters to the morphology of the tissue that you're trying to deal with. And so that needs some additional work. While we have a box of a laser that's about the size of 2 laptops, and that's a big improvement from where we were, this is not a cochlear implant. And we're going to have to do something about multiplexing. How can we stimulate at multiple channels simultaneously? So a little bit on that, where is that going? There is now built, again by Lockheed Martin, this device known as the Cat Pack, or the Rat Pack. This thing is about the size of a small iPhone that can be implanted underneath the shoulder blades of a cat. You can run the fiber optics to the cochlea, and Klaus is actually doing experiments in these animals, and hopefully he will have the results for that before too long. The next version of that, this is nice, but it's still too big. We can program this thing, by the way, transdermally using a simple PDA. Here is the next version of this. This now is a single diode laser that basically does the same thing as the box I just showed you a while back. And so this thing is now about 1 centimeter, sugar cube size sort of. And this already exists. And then the next generation of that is basically this thing here. So this is 1 millimeter by 1 millimeter with, I forgot the exact number, but about 64 or so individually addressable lasers on a single chip. So now you're going to really start, and so this is a VCSEL technology. It's basically growing these laser arrays on the chip. We don't quite get the power that we need and the wavelengths that we need, but in principle this is possible. And so I think this might really be the future of where a lot of these miniaturization and multiplexing efforts will have to go. I really want to talk about this aspect. Can we combine electric-optic stimulation? So what I've shown you so far is that we have what we think is an attractive alternative to electric stimulation for the reasons I mentioned. I'll come back to this graph, and I alluded to it earlier, and that is we can stimulate here, but we get damage over there. And I really think that this gap needs to be bigger. We need to have a larger safety margin to be able to do that. So the question is then, how can we either stimulate more effectively, or how can we reduce the damage threshold levels? One of the two will do the job. And so this is kind of what I was talking about. If you have single pulses and you wait long enough between pulses, and this is at 2 hertz, the temperature it's cooled off before the next pulse comes in, and life is good. But if you ramp this up to 5 hertz, you get thermal superposition, and this can potentially cause problems. So we figured, okay, well, how can we possibly bring the optic stimulation threshold down somewhat? So this is where the idea came about to combine this with electric stimulation, and we hypothesized that if we can prime this tissue with an electric pulse that is subthreshold, subsequently or simultaneously then dumping in a little bitty bit of laser energy to push it over the edge across the threshold, in theory we should be able to then dramatically reduce the amount of laser energy that's necessary to do that. And the benefit of that, of course, is twofold. It would allow us a larger safety margin. It would allow us to go to higher repetition rates without paying the price of possible damage. Facilitates multiplexing. This one is a big one as well. It's not only on the tissue side, it's also on the laser side. If I can get away with having to generate 1/10 of the power, it is much easier to build these devices, make them smaller, and deal with the device issues itself. So we did some experiments with that to sort of show proof of principle of that, and effectively really all what we did is take an electric stimulator, take our diode laser, bring both of them into the same nerve, had a delay generator to coordinate the timing between these two things, and basically, and here's the parameters for the laser, for the electric, and I'll just get to the results because that's the interesting part. So basically what you see here now is, as a function of the electric stimulation threshold, we determined how much optical energy do we need to throw into this tissue to get it to stimulate based on some sort of an endpoint, which in our case was a muscle twitch. So there's 2 points that you know a priori. You know that at 100% of electric stimulation threshold, at or right above the threshold, you don't need any optical energy. Conversely, at 100% of the optical energy, or right above it, we don't need any electrical energy. And so basically, the data points here kind of fall off that way. And what you can take from this is that, if we prime this tissue with a pulse that is at about 90% or so of the electric stimulation threshold, we can reduce the optical energy required to stimulate by probably about a factor of 4 or so. And that is very much in an unoptimized situation. We have hook electrodes underneath, the fiber coming in from the top. So spatially, they're not optimized. Temporally, they're also not really optimized. We so far have really done most of the work here. We're just giving both pulses exactly at the same time. And there's some suggestion here that we might be able to do better with that. So I think this can be further optimized, but the proof of principle is there that there is a superposition effect of the optical and the electrical. The other thing that's interesting here is that my simple-minded optical engineer brain would have predicted that this was just a linear superposition. The fact that it is not suggests that the mechanisms of stimulation are not the same, that they're targeting different things. So there's some other potential interesting issues to that we need to further explore. The spatial selectivity is maintained. This is a compound muscle action potential from one of the muscle groups downstream, and the other one stimulated doesn't show any signal as shown over here. So what you lose, of course, in this scenario is the artifact business, because now you have stimulated with a significant electrical pulse, even though it's subthreshold, you do pick up the artifact. So for that, it is not particularly conducive. But for the practical device-based approaches, it might be an approach to further explore. Okay. See how we're doing on time here. I've got a couple more things. So basically, I think we've shown that the electrical priming actually does lower the optical stimulation threshold. Again, they don't follow a simple linear superposition, and I'm not sure why they should, or should they. And so we're now working, and this is actually stuff that we've been working with Dustin on, to try to come up with some way of integrating a probe to combine these things in a spatially optimized fashion. And I want to leave some time for questions here. So I'll just get to some what I think are opportunities and challenges, and give you a little bit of teasers of some stuff that we've done and shown without having explored it extensively yet. To Towards human applications, I'll show you a couple of slides on that. The optical pacemaker, the biological mechanisms, of course, this is an area where I think we need people other than the optics folks to really start looking at it. I will say that, you know, working with someone like Hillel Chil, who's got his aplesia models and knows a lot about the neurobiology, is starting to get us in that direction. And I think that's going to be a critical part, not just from a scientific, you know, question point of view, but from a device development and optimization point of view as well. Another thing that has become obvious, painfully obvious to us, is that we need better recording methods. It's not so difficult in the periphery where, you know, you have that cable, and if you stimulate here, sooner or later that signal will go down that cable. What I won't talk about really is the central nervous system stimulation, which we've also shown so far. We can stimulate directly on the cortex and the cerebellum. The problem there is we now have such a high spatial precision stimulator, it is very difficult to figure out, you know, where do these signals go. We don't really I don't really understand or know the networks, and most people don't know exactly what's connected to what. And so instead of the sledgehammer of a big electrode and just stimulating everything, which you can see quite nicely, we're now touching this tissue with a feather effectively. And so we're finding ourselves sitting there time and time again where, you know, if we have an electrode in that same tissue to record something and we don't see signals, is it because we're not seeing it because we're looking in the wrong place, or is it because it's really not there? So we need better and more sensitive methods of detection, preferably being able to do full field mappings. And so we're actually exploring or moving in that direction and setting up a bunch of optical ways of trying to do that, which sort of makes the circle round. If you think about back to the beginning, how this whole project came about in the first place. You know, can we detect optical or electrical activity with light? And so we're now going back to that simply to facilitate the other part. I think there is some real work that still needs to be done on devices, miniaturization, multiplexing. The chronic studies, this one in my mind is a big one as well. I really strongly believe that the whole field, the interface between neuroscience, neurotechnology, and biophotonics is an up-and-coming field. I mean, all signs are there to suggest that that's indeed true. And not just our work in the nerve stimulation, there's many, many other things going on in that whole field. And so I think it is critical as a community that we really start to develop training programs that is going to produce people who are well versed in the knowledge and the techniques in the lab in both the neurosciences, electrophysiology, as well as the biophotonics. And that has to be more than just, you know, a person from one area taking the course in the other area. So I think it's really an opportunity, and I think it's going to take interactions between labs like ours and Hillel's to make that sort of thing happen. Okay. The last two little things I want to talk about, getting towards humans. So we actually have done this in humans. It's an initial pilot study that's still ongoing, but basically what we did here is we wanted to come up with a way of validating the experiments we did in the rat, proving that we can stimulate, that we maintain spatial selectivity, and that we can do this safely. So the procedure that we decided to use this on is a dorsal rhizotomy. And I don't know if you're familiar with it, but it's basically children with very severe cerebral palsy that go into surgery, spinal cord surgery. They instrument the lower extremities with EMG electrodes. They put a ball electrode on the dorsal rootlets, they stimulate, and they see which wires are essentially overactive and misconnected, and then they physically cut them. It's a very crude way of doing this, but that's the state of pediatric neurosurgery. What that gives us is an opportunity to come in with, after they've done the electric mapping, so the ball electrode stimulation, the EMGs are recorded, the ones that they decide are going to be cut before that has actually done, we come in with our fiber optic, stimulate, have the EMG recording, and then instead of doing a single cut, which is standard, standard practice in the clinic, they actually make a double cut, and we are able to take that little piece of tissue out for histological analysis. So that's the protocol. We've enrolled, I believe, 7 patients in this to date. It's not a common procedure, so we've only done, I think, it's taken us 2 years almost to get 7 patients in that study. They all happen in the summer because there's a pediatric population, so they happen early summer, so these kids can recover over the summer. But it's slow going. But that's basically the procedure. And I'll show you one example. It's a little dark slide because it's a screen capture, but basically this was the electric ball electrode stimulation on the dorsal root. In the blue here is the left leg, and the red here is the right leg, and each trace here is an EMG electrode. And what you can clearly see here is that this is a crazy going nerve with all sorts of ringing and feedback and overexcitability. And so this is one that clearly needs to be transected. If we then come in with the optical stimulator, and it's really hard to see, but you see nothing in any of these traces here. The only one that you see something on, I'll follow you along here, is one nice action potential trace in one of the EMG electrodes, suggesting that we very much maintain the spatial selectivity we were talking about before. And remember, this is just about the worst possible case you can have, because these are, I mean, you look at these neurons the wrong way and they start firing. Histology on that we have, but it's not been analyzed, so I won't be able to show you that. But the initial indications are, not surprisingly, is that we're not causing damage with the same parameters that we also were not causing damage in a rat. So, so it looks pretty good. And I wanted to throw this one in here because I think it's a really exciting little project. We just started doing this last summer when Melanie Gold was here in Austin, Duke, one of my grad students, started to work with, with Mike Jenkins and Andrew Rollins. So they're doing all this work on the embryo, the heart development in the embryo. And so here we actually decided, ask the question, can we stimulate the heart with light? So basically this is sort of, to my knowledge, the first ever proof of an optical version of a pacemaker. So we have a laser Doppler probe to actually measure the flow here. Here's the heart. And this is a quail embryo. I forgot exactly how many days, but the laser fiber is coming in here. And when I turn this on, what you'll see is the heart is just pumping and doing its thing. And the moment we start turning on the laser, it synchronizes the heart and starts pacing it at the rep rate of the laser exactly. So here it's just going happily on its own, and then the red line here, the laser comes on, and we start triggering it. And you turn it back off later, and it goes back to its own nascent state. So we're further exploring this and optimizing this, and there's all sorts of interesting things that one can potentially start to do with this. So. All right, so just to wrap up here, I hope I've convinced you that optic stimulation presents a relatively simple but novel approach that gives us this ability to stimulate excitable tissues in a contact-free, damage-free, artifact-free, and spatially highly specific mode. We validated this in a number of different systems, both peripheral and central, motor and sensory. I've mentioned this explicitly, but it is critical to realize that the optic stimulation wavelengths, and in fact all the parameters, but they have to be matched to tissue morphology. Just what works in one nerve does not necessarily optimally work in another type of system. And so this is something that is a lesson that we learned, and many of our collaborators and other people starting to do this are figuring this out now as well. So coming to my acknowledgments, clearly this is a lot of work by a lot of different people, both at Vanderbilt and our grad students. They're really the guys doing all the work. Sharon Thompson, who's the pathologist who works with us. The guys at UT Austin who helped us with the differential phase OCT. Big part of all the cochlear work really is done at Northwestern. Lockheed Martin Acculite is absolutely crucial for us to move forward with the instrumentation development. On the central nervous system, Anna Rowe and Bob Friedman at the psychology department. Here at Case, Hillel, Andrew, and Michael have been great collaborators to work with. Early on, our free electron laser takes a lot of people to make that thing produce photons in the first place. Unfortunately, that program was cut about 1.5 years ago, but I mean, I can honestly say this is one project that having that FEL was critical, and it probably would have never happened if we hadn't had that project or that program in place. On the funding side, these are our funding sources, and I think with that, hopefully there's some time for questions. Thank you.
much. So we have some time for questions, and for those that may be watching the video feed, we have a couple different places where we can get email, so please send your questions in, and we will try to get them out here.
I just did a quick,
Hold on, I'll give you the mic for the video.
so you did
The piezoelectric mechanical stimulation to sort of, I was curious, since you're targeting possible like a thermal effect, did you do a thermal-based probe not coupled to light to see if you get a similar sort of?
We haven't actually done that. So the question, if I understand it right, is you know, can you use like radio frequency or something else to just transiently heat that material and get the same effect? My hunch is you will be able to. Getting, you know, so why do you need light to do this? And the beauty of you know, light specifically is that you have this very fine ability to tune to a wavelength, and it gives you the volumetric energy deposition. Most other methods, whether it's a conduction method, you know, you're depending on thermal conductivity and the like. If it's something like radio frequency, you know, it's much more of a bulk heating over a much larger volume, and you don't quite get the gradients. So the beauty of doing this optically really is that it allows us to pick the wavelength, and you know, it gives us almost 9 orders of magnitude of dynamic range in terms of the energy penetration depth and where we want to exactly deposit that light in that tissue. My gut feeling is if you had enough other way of depositing the light or depositing the heat spatially that way, that will probably work as well, but we haven't actually done that.
I was struck by the difference in the compound action potentials that you got from electrical stimulation versus light stimulation. And there is, I imagine, some difference in how much myelin is on the nerve fibers in a fascicle, and whether or not myelin or the structure of the nerve fibers that are being activated play a role, and whether or not you have looked at recruitment order and seen if lightly myelinated axons are recruited before a myelinated.
We have not systematically looked at the amount of myelinization, but we have looked at the recruitment, and what's clear from the experiments and everything we've done so far is that, again, this goes back to mapping the optical penetration depth to how many axons do you actually recruit. And so there's a, you know, if you dump that light in 50 microns, you're going to recruit 50 microns worth of axons. And if you dump it over a spatial range of 200 microns, you're going to recruit 200 microns worth of axons. Electrically, you don't have that, you know, that ability to do that. So, which is, I mean, that's the primary reason why the amplitude is so different, because we are recruiting much fewer axons. You know, if you go to some other wavelengths, which I haven't shown, we also have another version of this laser that if you remember that absorption peak, that sits right at the peak. So now we have a penetration depth of like 40 microns. So that's good and that's bad. It's good in the sense that it allows us to now only activate 40 microns worth of axons. It's bad in the sense that, you know, we still got a 200 microns worth of connective tissue on the outside to get through. And unless we then physically remove it, we're never getting the light down to the axons in the first place. You know, I'm hopeful, and I think the way to go with this is to try to not have to start poking things in the nerve. That would be, and you know, we had some conversations about it at a conference call the other day. I don't think it's impossible, but if we can stay away from that, I think from a practical point of view and clinical point of view, I think that is highly desirable. So that automatically means that we got to pick wavelengths that at least get us to these, you know, to the connective tissue on the outside. And of course, if we go to humans, you know, we're going to have to scale that accordingly.
What's the maximal frequency of evoked potentials you can obtain in the nerve without the thermal effect?
Well, it depends in what nerve you're talking about. What I've shown you is in the periphery, where we have significant amounts of energy that we need to throw at this tissue simply to get through. Think of it this way. In the rat sciatic nerve, we're throwing a lot of light away that never gets to our target. And so there, I think with the parameters we've done so far, I would be hesitant to go much above 5 hertz, which is low. What I've also shown you, if you go to the cochlea, where the thresholds are 2 orders of magnitude lower, therefore reducing the heat load, now you can get away with, you know, a couple of 100 hertz easily. And you know, at some point you start dropping pulses, but that has nothing to do with the heat. necessarily. But so it really depends on what the specific structure is that you're targeting and how effectively you can get the light to your target tissues, target cells, and hope that answers the question. So, yes.
There was a recent study, maybe about a year ago, out of Stanford, looked at mechanisms of deep brain stimulation using optical stimulation. A lot of the data that came out of that flew in the face of probably several hundred manuscripts published on mechanisms of stimulation and pathophysiology Ph.D. Can you comment on what you think some of the issues might be with that study, or how we might be able to reconcile what they talked about versus what a lot of other people are talking about?
I'm not sure. I'm not familiar with that paper, so I'm hesitant commenting on that.
What if I send it to you and maybe
that will be? I'd love to see it. Absolutely. Yes. Yeah. Were they using what were they using? Do you remember? What's up?
I was going to read it before I came over here today, but I didn't get to it. But I'll get your email address.
Absolutely. Yes.
I actually have a question. You mentioned selectivity being vesicular selectivity, two different muscles. Do you think that this can go beyond that to subvesicular, or what do you think the limits are as to how small you can make that spatial resolution?
There's two answers to that question. I think sort of fundamentally limited are fundamental limits are things like diffraction limit of light. We should be able to get to microns, actually, and microns laterally. If you pick wavelengths that are strongly absorbed, we can deposit that light over a micron. Actually, we can go to in the axial direction. We can go to a few microns. If you go even fiber optically, I mean small fibers, single mode fibers, you can easily go to 4 micron, 5 micron fibers. So from that point of view, in terms of what volume can we interrogate, I think we can do single cells. I'm convinced we can do that, at least in the Petri dish. Now, the practicality, of course, and the hard part is how you're going to do this in a bundle, that's a millimeter or a centimeter in diameter, and how you're going to get things in there. But this is where it's going to require some out-of-the-box thinking, and maybe we can get fibers in there, or maybe we can stick something on specific tissues that cause the light to be absorbed, so we can use wavelengths like the alexandrite, the red light I showed you. I mean, red light is not absorbed in tissue. I mean, if you take the red laser pointer, I do this in my kid's 7th grade class also. It's no, it's not. No. Look, all right. So if you take red light, you can probably see this. Light goes through tissue beautifully. Red light does. Now, if I did have a green light, it doesn't do that. Okay. So what does that mean? It means that this wavelength isn't really absorbed very strongly. Everybody's done this. Take a flashlight, stick it in your mouth, stand in front of the mirror, and look funny. What do you see? It's red, right? So the red light, the red part of the spectrum, is not very strongly absorbed. So what that suggests is I can get red light very deep in tissue, but now it's not absorbed. So what if I stick something on these nerves, a nanoparticle or a gold sphere or something that is absorbing that? Now I can enhance the selective absorption locally. We've dabbled into that a little bit as well. So I think, honestly, I do think that the fundamental limits are single cells. I really do believe that. The practicality of that in vivo, there's challenges there. But certainly in the culture, the work that we've started to do with Hillel, I mean, we're stimulating, of course, they're big cell bodies, but we've already done this. We're stimulating single cell bodies in the aplysia prep. And they're about 100 microns or so in diameter. So, not quite at that level, but I think that's possible.
Any other questions? My latest check on email. Okay.
So you can activate single cells, but that's, as you showed in your X potentials, that's much lower. You get much lower force because they're activating fewer fibers. So that's all right. So once you can activate fewer fibers, you also need to be able to activate more than that to get something functional. So, but you can't. Your lasers are not very modulatable in the frequency of the light right now, right?
Correct.
So how do you get? So one thing is to get very little bit, but how do you get enough to get a functional movement out of it?
There's a number of things you could do. I mean, I'm completely speculating here. there's two aspects to it. One is the multiplexing. How do you stimulate multiple cells at the same time? And so it is actually not that difficult to do. If you have, think of it this way. If I take a very small fiber, okay, I can either throw all my light in a single fiber, or I can make a bundle of these fibers. I can pack them. And this is what's done in endoscopes all the time. There's thousands of fibers in an endoscope. So if I can just throw that light at all these fibers at the same time, and at the end I can kind of, in theory at least, start to aim these different fibers at different cells. In principle, at least synchronously, I could stimulate a whole bunch of them. I don't think that would be too hard. What will be difficult is if I want to stimulate them sort of in a temporal pattern, then you're going to have to start resorting to perhaps optical switching. And again, a lot of that technology exists because of the telecom industry. I think they're overcomable technical obstacles there that can be solved. We haven't done that, but I don't see any fundamental obstacles to try to get there. But keep in mind, I mean, this is a big tree with a lot of fruit on it, and we went for the low-hanging ones first. There's a lot more stuff that needs to be done. There's a lot harder problems that still need to be solved. But we figure we'll just do the easy stuff first, and then we'll worry about the harder things later. Which is always a good idea in the lab anyway.
Any other questions? With that, we have a small token of our appreciation for
thank you very much.
Providing the lecture today. And thank you. And thanks everyone for coming. I'd like to remind you one last time, the next speaker will be Doug Weber on the 19th of March. So thank you very much.